Good morning, everyone. So, we all know this is a fabulous meeting. We all enjoy it. This is my 4th time at ICMS, and it's always great fun. It's great fun to meet old friends, or whatever you want to call each other, and also make new friends. So, this is a fantastic meeting, and we've all said thank you to Sophion, but I'm going to say it again. Sophion, you're doing a great job. Thank you very much. And I'd also like to thank the organisers for allowing me to present some data. Now, this is my title, KV7 Channels Go Cycling. This is not a nod to the Danish passion for getting their bike out and trying to run you down as you cross Copenhagen Street. This is actually a summary of about 4 years' worth of research looking at the impact of sex on KV7 channel activity and the effect of modulators of these ion channels. So what I'm going to do today in the next 20 minutes is I'm going to provide an overview of these ion channels, because not everyone is in love with these things as much as Jeff and I are. We absolutely love KV7 channels. So I'm going to give you a background, and then I'm going to present the view of the newest sort of cell type, the smooth muscle cell, and its relationship with KV7 channels, expression profile and other effects. And then, in the middle bit, I'm going to present some data that was published last year that stemmed from the PhD work of Sam Baldwin, who's now doing a postdoc in Sydney, where he was looking at the impact of sex and the estrous cycle upon KV7 channel regulator activity. And then I'm going to end with some more preliminary data generated by a final year PhD student, Zaina Weber, who is in the audience. So if you have any questions, don't ask me, ask her. She's at the back. So let's get to it. So this is the very simple overview and summary. So there are 5 KCNQ genes, 1 to 5, that encode these proteins, KV7.1 to 7.5. 7.1 was identified in around about 1996, and KCNQ5 was identified in the year 2000. These proteins form tetramers to create voltage-activated potassium channels. You don't need much depolarization to get these channels to open, and in general, the general dogma is that KV7.1 tends to form homotetramers. It holds hands with itself, whereas the other KV7 channels can form heterotetramers with preferential associations. So 7,2 with 7,3 and 7,4 with 7,5, other sort of common combinations. There are a wide range of things that modify KV7 activity. Lots of work done by Jeff Abbott over there, and other people have identified, and we heard work yesterday from Nakajo about KCNE expression products. So these KCNE gene products alter the trafficking, biophysical properties and pharmacology of KV7 channels. Recently we identified, following on some work again from Jeff, this seems like a bit of a promotional tour for Jeff Abbott at the moment, we've identified that inositol transporters also associate and shape the activity of KV7 channels. And it's been well known that local phosphoinositide levels regulate KV7 channels. And about 8 years ago, we identified that beta-gamma G proteins really regulate KV7.4 activity. And there's a range of pharmacological agents stemming from the fact that an activator called retigabine was a very effective anticonvulsant agent. It got withdrawn from the market because it turned you into a Smurf, turned you blue, a photolytic product. So this is the general accepted expression profile. This is taken from a review article, and I've just added the newest thing, the smooth muscle cell. You can see that these are the proteins up here and the genes associated with them. And basically, KV7.1 is found in cardiac myocytes and also in lots of epithelia, including in the inner ear. And mutations to KCNQ1 lead to hereditary arrhythmias, hereditary deafness, etc., etc. KV7.2, 7.3, and 7.5 to some extent. 7.2 and 7.3 are found predominantly in neurons, and mutations to KCNQ2 and 3 underlie a number of hereditary epilepsies. And you can see in this diagram that KCNQ4 is found in the inner ear, and you can get some hereditary deafness due to mutations to KCNQ4. But also down here in the smooth muscle world, which is what I focus upon, we've identified that smooth muscle cells, both vascular and non-vascular, express KCNQ4 and 5 at higher levels, and there's also a contribution from KV7.1. So this is our expression profile, and I can explain a little bit more if you need. This is our general pattern. And a very, very simple summary. So my labs looked at lots of different arteries, as are the other labs around the world. And you can see all these arteries here. They all become more contracted. That means more narrow if it's an artery. If you give an agent such as linopidine, that blocks all KV7 channels. So that makes sense because these channels are keeping everything calm. And if you therefore disable them, either pharmacologically or we've used molecular approaches, molecular interference, you get a more contracted artery. And there's a little experiment that we did. This is a renal artery. Black is a control response to an alpha-adrenal receptor agonist called methoxamine. And this is repeated in the presence of a KV7 channel blocker. And you can see the starting point's higher because it's already contracted in response to this agent. And so there's all these arteries, aorta, coronary artery, cerebral artery, mesenteric artery, penile arteries, etc., from different species, including our work in human arteries, all become more contractile when you inhibit KV7 channels. And conversely, agents like retigabine that activate KV7.2 to 7.5 relax pre-contracted arteries. So here we've just we've contracted the artery so it's narrowed, and then we've added 2 concentrations of this lovely drug, acrylamide S1, and we get a lovely relaxation. That is reversed by a KV7 channel blocker, XE991. So you're all happy with this situation, that KV7 channels are really quite important in arteries, keeping things calm, keeping them relaxed, and if you disable them, your artery becomes more contractile, it becomes narrower, and if you enhance them, you can get vasodilatory responses. And then we've also shown that these ion channels are very important for many endogenous receptor-mediated vasodilators to work. So if you disable KV7.475, either pharmacologically with blockers or by molecular interference, and we've used siRNA and also translation blocking morpholinos, we've got them into arteries. If you do that, then a whole host of receptor-mediated vasodilators like adenosine, CGRP, or beta-adrenoreceptor agonists don't work so well. So all these agents work through a G-protein coupled receptor that leads to an increase in cyclic AMP. I don't know whether there's a cyclic AMP sponge involved. I just learned that yesterday, so that's great. I might look for that. But they can signal either through protein kinase A or EPAC, exchange protein activated by cyclic AMP, and there's some sort of activation of KV7 channels. So I'm just going to leave it there. But also, interestingly, atrial natriuretic peptide, which promotes an increase in cyclic GMP, also relaxes arteries less well if you disable KV7 channels. So, in summary, KV7 channels, very important for resting arterial function, but are also important for a lot of receptor-mediated vasodilatations. They're not the be-all and end-all, but they do contribute quite significantly to different responses. So that's the situation in males. What about if your research animal happens to be a female? And we're working mainly with rats. And this is where Sam started his PhD. And for those that don't know, the rat has an estrous cycle of about 4 days. We have these different stages here. And what we do is we have a rat, we take a vaginal smear, we look at the cytology, and we also look at the state of the uterus. And that kind of allows us to stage where the animal is before it was culled. Okay? And this is taken from a textbook. We also have some data from a reproduction HPLC unit at the University of Edinburgh. And you can see there's changes in estrogen and also progesterone, but there's also some other hormones going on. It's very complex. But what you're going to see in a minute is I'm going to show you data in arteries from these different stages. And you're going to see red lines and blue lines. The red line is in this stage, the proestrus and estrus, which I might abbreviate to P and E if I'm being lazy. That's a sort of a higher estrogen level. There's also some progesterone, but we're going to focus on this. And then I'm going to show you some blue lines, blue boxes, that are in these other stages where it's a lower level of estrogen, okay? So, in these experiments, we've mounted an artery on what's called a wire myograph from another excellent Danish company, Danish Myograph Technology, from Aarhus originally. And we measure the force associated with these arteries trying to narrow. And you can see here, you add a vasoconstrictor, it contracts, it narrows, so this is an increase in tension this way, and then we've added a Kv7 activator, 7275 activator, this one's called S1. And you can see concentration-dependent relaxations, and here's the mean data from many experiments shown here. So you're all relatively happy with that, aren't you? It relaxes, yeah, it's not super potent, but it relaxes quite nicely, nice error bars, thank you very much. And this was from rats that were in this red box stage, the sort of higher estrogen level, okay? Are you all happy with what you're seeing so far? Do you understand what you're seeing? Yes. Thank you. Because the next slide, I'm going to show you data from the rats that were in these stages. So they're very different estrogenic or hormonal state, and let's see what happens to the Kv7 activator response, because it's quite amazing. Massive increase in effectiveness of this agent to relax these arteries, which were renal arteries. You can see the response here, and you can see how tight the error bars are as well. There's very little variation. So this was quite amazing. I actually said, Sam, you're an idiot, you've done it wrong. You can't do dilutions. And of course he did it again and again, and then I blinded him and everything. It's real data. And so we looked a little bit further. We looked for the same drug in different arteries. And again, blue is always the lower estrogenic estrous stage. We call it D and M, but let's just call it the lower one. And red is the pro-estrous estrous stage. And look at the difference in potency of this agent in coronary arteries, cerebral arteries, and mesenteric arteries. We also looked at a structurally different and still Kv7.23 activator called ML213. And again, the same phenomenon exists. So it's a Kv7 thing, it's not this drug. We also looked at the effect of a blocker in these two types of arteries. And you can see the blocker, line opening, blocks all Kv7 channels, produces a far bigger contraction if the artery is from a female that's in diestrus or metestrus compared to a female that's in proestrus and estrus. And we looked at vasoconstrictor responses to thromboxane, methoxamine, vasodilator responses. There was always this thing that if the animal was in this red stage, the higher estrogenic stage, you got a more contractile artery than if it was in this stage. So what was driving all of this thing? So obviously initially we went to transcript, quantitative PCR. What about the KCNQs? What about the regulatory proteins? But to cut a long story short, not much difference in the two female groups, so within the estrous stage. What about the localization of the protein? So we skipped the Western blot, we went to immunocytochemistry, and we used two detection methods here. The green is wheat germ agglutinin, so that gives you a membrane localization. And this is a cross-section from a confocal microscopy experiment. And you can see that Kv7.4 targeting antibody, shown in red, you've got some membrane abundance, but of course there's going to be protein in the cytoplasm as well. And then when you take an XY plot, shown by the yellow line, you can see that there's lots of Kv7.4 in the membrane, overlapping with the WGA. That's in a D and M, the blue box. What about in the red box, as we see here? And you can see that there's very little Kv7.4 in the membrane. It's translocated. And remember, the estrous cycle of a rat is 4 days. So you're not talking about long-term effects. It's happened maybe in a day or a couple of hours. So it's moved out of the membrane, is now more cytosolic. Here's just the mean data, lots of experiments. And we also looked at 7.1, 7.5, and also KCNE4 encoded proteins. And you can see that there's really no difference in the membrane to cytosol ratio, i.e. there's no difference in where the proteins are, whether they're in diestrus, metestrus, or proestrus and estrus. So it seemed to be just 7.4 that was moving. Okay, so what's driving this? Now, I'm not saying, of course I'm not, that there's only one hormone involved. There's lots of hormones. There's progesterone, follicle-stimulating hormone, luteinizing hormone, and estrogen. But we just focused on this one because the data that came from Edinburgh showed that there was a change in the plasma, detected in the plasma from the rats we were working with. So let's have a look at estradiol. And you probably all know that estrogen works on these, let's call them canonical receptors, these steroidal nuclear receptors. And classically, most textbooks would say that these estrogen receptors tend to affect gene transcription. The estrogen will bind, get to the nucleus and do some magic. But ER alpha is also known to exist in the plasma membrane, and it can have very acute effects, very quick acting effects. And there's also this recently identified, I say recent, it was about 20 years ago, now called GPER1. That is a G protein coupled receptor that is found in the plasma membrane. And there's also a relationship, depending on the cell type, between these two types of receptors. So what might be driving these estrous cycle changes that we see? So we've got some data now. It's blue, so it's females that are in diestrus, metestrus, and we're just going to add exogenous estradiol to see if we can mimic our estrous cycle effects. So we add estradiol, E2, 10 nanomolar, nice sensible concentration, and it produces the shift we see with the estrous cycle. Okay? So we've kind of mimicked what Mother Nature has given us. But which one of these receptors is doing the effects? So we focused on this one because this was only 30 minutes of estradiol. That's not really long enough if you're working on a textbook to change gene transcription. Those gene transcription changes then manifest as protein changes, and then they change cellular responsiveness. So we thought it might be this GPER1 that's doing these things. And there is a specific agonist of GPER1 called G1. We did the experiment with G1 incubated beforehand, and we saw very similar, if not identical, effects to that produced with estradiol. So a shift in the responsiveness to ML213. We also identified, or there is a blocker of GPER1 called G36. And when we repeated the experiments with estradiol, E2, but now in the presence of G36, we didn't see the impairment. So it's kind of pointing the finger at this receptor. When we then look in P&E and we do the enrichment experiment, 30 minutes of estradiol, we see no effect because it's already, you could say, affected by the estrogen floating in the blood. Males, we did the experiments as well in arteries for males, and we saw a little impairment because there are estrogen receptors in the smooth muscle of the males as well, with both estradiol and G1. So that's nice. We've implicated GPER1. And we all should acknowledge past work. It's very naughty when people don't do this. It's very upsetting when it doesn't happen. So Brian Harvey did a lot of work before he switched alliances, and then he's now working in astrophysics, for those that don't know. And he did a lot of work looking at the effect of estrogen state in colonic cancer mechanisms. And here they've shown that KCNQ1 binds beta-catenin, and then with estrogen you get an uncoupling from the KCNE protein. It allows the Kv7.1 to sort of float off. The beta-catenin loses its anchor, and then the beta-catenin goes to the membrane, and it drives this epithelial to mesenchymal shift. And they were the first to suggest that there was an acute effect of estradiol, not this long-term effect. So that was kind of driving our thoughts, and I just want to acknowledge this work as I get back to our work. So with that in mind, we then thought, okay, well now we've seen what happens in cellular distribution across the estrous cycle. What about in our estrogen-enriched experiments? So again, we've got our confocal microscopy, we've got our membrane marker, and we've got our ion channel shown in red. So in ethanol, it's in the membrane mainly. I've just truncated it. Then when you give estradiol, you can see that after 30 minutes, the Kv7.4 has flipped out of the membrane phase, just popped over a little bit. We also saw a similar thing with G1. We also got some cytosolic accumulation. And these responses were prevented when you had the GPER1 receptor antagonist. So we've kind of shown with imaging techniques what we also saw in the functional activity, that there's this GPIL1 receptor mediated flipping or translocation of Kv7.4 from the membrane. Okay, so it's an ion channel meeting and there's been no current so far. That shows an index of about my research, to be honest. So this is the last electrophysiological data that my lab generated, and this is about 3 years ago, so sad state of affairs, but never mind. And what we've got here is a HEC cell line stably expressing Kv7.4, and then we transfected in a GFP-tagged GPIL1 receptor. So the experiment was patching a cell that was glowing or patching a cell that wasn't glowing, adding estrogen or G1 and seeing what happens to these Kv7.4 currents. So here you can see lovely voltage-dependent Kv7.4 currents. We've done all the characterization, and the red one is like a little marker. And this is a non-glowing cell, so it hasn't got the GPIL1 receptor. We then add G1 or estradiol, whatever, and you can see there's no real difference. Look at the reds, and here's the mean data for many cells. No effect, there's no GPIL1 receptor. That's great. Now we recorded from this cell here. There's a big red arrow to show you which cell we recorded from. Thank you, Sam. He doesn't think I'm an idiot, obviously. And here's the little IV, and here's the little marker current, and then we added G1. Ta-da! GPIL1 inhibits the currents within 15 minutes. Big effect. And here's all the mean data for G1 solvent, E2. Within 30 minutes, you get inhibition of Kv7.4. And I know the question you're going to ask, did you do the staining work and did the membrane abundance go down? Yes, it did. So how is it doing it? How is it doing it? How isn't it doing it? So it was only really Sam working away, bless him. And so we use this technique that we've used in the laboratory called proximity ligation assay. For those that don't know it, if the protein's within... is that it already? Good Lord. Okay, so to cut a long story short, we've already identified in the part the heat shock protein 90 associates and traffics Kv7.4 of the membrane. It doesn't associate so well in estrogen enrichment and all that type of stuff. So very quickly, and I'm going to do the next bit very, very quickly because Zane will be very upset. Kv7.4 is in the membrane at the low estrous stage. When you have the higher estrous stage, Kv7.4 is flipped out the membrane, and we've implicated a reduced association with this forward trafficking chaperone called HSP90. So very, very quickly, Kv7 channels aren't just important in arteries, they're also very important, and this is work that we've done and other groups around the world. They're very important in the uterus, airways, and bladder. And so Zane has been working on the bladder for a couple of years now, so they're functionally important there. Is there sexual dimorphism in the bladder? And in the bladder, it's spontaneously active, but also we can activate some nerves and you get these evoked responses. Here's our colour scheme again. Well, these are just basal properties. And you can see that the male has bigger contractions to nerve evoked responses, area under the curve here, but the spontaneous activity is higher in the females. What about the drugs? Little snapshot, representative trace. There's never enough of these in science, is there? So here we have the spontaneous activity here, nerve evoked responses here, increasing Kv7 activator there. Look, concentration-dependent responses. And you can see, without me having to show you mean data, that this drug is less effective in a bladder from a female in proestrus or estrus, but also in diestrus and metaestrus. And here's the mean data. This is the effect of NO2 and 3 on spontaneous responses and nerve evoked responses. And you can see that the responses are a lot more effective in the male. And this is slightly different than what we saw in the arteries. And here's a load of nice data down there. What about the enrichment study? So again, we take the bladders, strips from the different animals. We know what stage they're in. But then we added estradiol again for 10 nanomolar for 30 minutes. And you can see, here's the ML213 control response. In blue is the response to estradiol. If the rat was in diestrus or metaestrus, you got a nice impairment of the response. But if the rat was in proestrus and estrus, we didn't see this. Cellular distribution, I'm running out of time, but to cut a long story, very short, Zena has seen there's a difference in Kv7.5 localization between the estrus stages. There's more in the membrane and DNM than there is in the P and E. And finally, and this is the final slide, Well, she's been looking at the estrogen receptors, and she's also looked at the other ones, not just GPO1. And what we're looking at now is the response. These are sort of like the control experiments. This is the response to ML213 under control conditions, and then no added estrogen. This is just the receptor antagonist. And what you can see here is that G36, that blocks GPO1, actually produces a slight shift in the bladders from FDM, but a significant shift if the bladder's from P and E. So there's some endogenous activity going on. But the really interesting thing is with fulvestrant, which is a blocker of ER alpha. In D and M, there's not much effect of the blocker itself, but in P and E, you can see there's a marked enhancement. So there's some constitutive ER alpha and GPO1 activity going on in this stage that's combining with all the complexity. And we've obviously got a lot more to do, but that's where we're going to end this story. So, arterial smooth muscle cells respond to Kv7 channel modulators. That is affected by sex and estrous cycle. Similar issues occur, but with different mechanisms in rat bladder, and the underlying mechanisms need to be defined. Whee! Let's thank all the people that have put up with me over the years. My very first postdoc, Shuki. Oh, lucky person, never mind. Sam did all the work, Zaina's done the most recent bit, and these are the people that are still in the lab. And collaborators, lots of lovely people doing lots of lovely things, and the occasional, sometimes, funders. That's the way I said that. Thank you very much for your attention.
Thank you very much for the great story. Sorry for
That's all right, it's okay.
picking you up.
I've had many people get up and leave.
Okay. We have many questions, so we
Oh,
start
wow.
from
This is working.
left and we go right.
Hi, thank you very much for the talk. I was just wondering, so for example, where you have those on the combined contraceptive pill, where you have higher estrogen and progesterone levels, you get a higher incidence in increased blood pressure and blood clots, I think. Do you think, amongst other things, this could be due to an increased sensitivity to estrogen in Kv7.4 channels in these patients who experience this?
That's a really interesting question. So the question there is about the mixed contraceptive pill and the associated problems. We're just taking a snapshot, and even when you're looking at the estrous cycle, there's other hormones going on. But that is indeed something that we would think contributes to. And again, I am not one of those polarized scientists who say, my research is the only way that happens. It is well known that estrogen can affect endothelial-derived mediators and lots of other things. So this is just one part of the story, but yes, it leans that way. But also, interestingly, it's well known that some women experience menstrual migraines, and that occurs in the low estrous stage, and we think, we've got poor old Sky's now research looking at this, we believe that would be when the Kv7 channels are more active in the cerebral arteries, and you've got a more dilated response at the low estrous stage. And that contributes, that's what Sky's working on, a PhD. I hope that answered your question.
Hi, my questions are about how this is conserved in the human as a mechanism. And a follow-on question, I guess, is do we need to reevaluate the adverse effects in clinical trials with reticabine that identified urinary retention, and whether we need to reinterpret that in the context of the estrous cycle?
I'm going to answer the second question first. What this data shows is buyer beware. If you look at those concentration effect curves, if you give 1 micromolar ML213, you are hearing bladder activity, and this would have happened. Retigabine is not as effective as these agents, but yes, you have this urinary retention. In a male, 1 micromolar, you've got full bladder inhibition, and you get urinary retention, and that's great. 1 micromolar in some of those female groups, you've got very little effect on spontaneous activity at all. So you've got this window where if you're trying to treat someone, you might get no effect or you might get very big effects. And therefore, we all know that we have to take on board the sex difference in our research, but this would suggest it's very important in clinical targeting or that. As opposed to re-evaluating its effectiveness, I don't know how estrogen affects the neuronal Kv7 channels. Maybe other people do. And in terms of its side effects, there might be issues there, but it was removed from the market because of its phototoxicity effects and other things. The first question was?
Well, just how we know the mechanism.
Yes, so the question is, so how about humans? I've just been able to get my ethics approved to get human arteries again, so we're going to be starting getting human arteries. But weirdly enough, people don't want to give their mesenteric arteries to me anymore. I don't know why. Medical students are so fickle these days. We have another question?
Yeah.
I have a question about the coming and going of the channels into the plasma membrane.
Yeah.
Do you have the idea that they are sort of going forever, or are they recycling, and what you see is basically just the imbalance of a coming and going?
Super question, and I would like to answer that, but I haven't got the personnel. It's usually just Sam on himself doing it. It's now Zaina just doing it. So the simple way would be to tag it in an expression system and follow it round. I think it's just constantly cycling, and we've got there is some hints from Tom Jeps's group in Copenhagen that the microtubule and dynein removes Kv7.4 and those type of things. We've shown that if you treat with angiotensin and upset the heat shock protein, which is a forward insertion mechanism, then the Kv7.4 gets transferred to a ubiquitin ligase and puts in the proteasomal bin. So this doesn't answer your question, but the answer is I don't actually know, and I would love someone to do that tagged Kv7.4 experiment and see it chase round the membrane. I think it just flips and back because it's just so quick. 30 minutes of estradiol, it flips out. You're going to say, oh, did you do the washout experiment? Damn, we should have done that. I don't know, did it flip back? But yes, I don't know.
Okay, thank you again for
Thank
the
you.
talk. And thanks for